Okay, boys, plankton breakover, pull your fish fingers out, come on. We have some scuba divers approaching. Now you know the drill. Everyone into position. Okay? 2, 3, 4. Da-da. 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 Louder, boys, louder! Give me fortissimo!
Chondrichthyes is a diverse group that includes the sharks, skates, rays, and chimeras. Many of the living sharks and rays are in the same genera as species that swam the seas over 100 million years ago. Sharks and rays are characterized by a skeleton primarily composed of cartilage. The majority of sharks and rays are marine fishes, but many enter estuaries, some travel far up rivers, and a few are permanent residents of freshwater. Most species live in the relatively shallow waters of continental margins or around offshore islands. A few roam far out in the vast spaces of the oceans. Some live at great depths in midwaters or on the bottom. Others are surface swimmers or inhabit the bottom and shallow waters. The smallest shark in the world is the dwarf LanternShark, and it is smaller than the human hand. These bioluminescent sharks get their glow from light-emitting organs called photopores along their belly and fins, which is used for camouflage from predators above them when they feed in shallow water when it is sunny outside. In darker water, the light attracts smaller prey. These characteristics could be the reason why the species is rarely ever seen. We do not have much information on the species as it has only been observed a few times.
3 male dwarf lantern sharks, Etmopterus splendidus, were caught and contained from the East China Sea off Okinawa Islands from the depths of 470 and 485 meters. When the sharks spontaneously lit up, photos were taken of the ventral and lateral sides to establish the order of the luminous pattern. At this time, a non-luminous band around the shark's infrapelvic and infracaudal zone was observed. This is referred to as the caudal dark band. They also observed 9 different luminous zones that were calculated to cover approximately 91% of the total non-translucent ventral surface area. After being sacrificed in the name of science, pieces of skin containing photophores were collected from different areas of the shark's body to determine the photophore density. Photophores are small light-producing organs that occur in a variety of marine animals, including fish and cephalopods. With gathered information, the researchers performed an analysis and determined that there were considerable differences in photophore density over the different luminous zones, from 3,900 photophores per centimeter squared in lateral luminous zones to around 8,600 photophores per centimeter squared in pectoral luminous zones. The average total photophore number was approximately 136,000 for the 3 sharks. It was observed that photogenic areas containing higher photophore density seemed to emit brighter light. In the caudal dark band, photophores were totally absent. Structure of the E. splendidus photophores was also examined. The photophores were made up of bell-shaped organs consisting of several photocytes covered in pigmented casings atop a lens cell. Photocytes are cells that specialize in catalyzing enzymes to produce light. This overall type of photophore structure is also found in other Etmopterus sharks.
To observe a physiological control of photophore luminescence, a screening of various purchased substances took place to identify the shark's photophore's ability to induce light emission and to later compare it to a previous study on E. spinax. Statistical analysis then took place to observe the results from luminometer readings and microscope observations on various luminous zones along the body of the shark. They found E. splendidus photophores undergo a complex physiological control by hormonal and neural inputs, which is similar to the other species of lantern shark, E. spinax. The comparative study found the 2 species of lantern sharks had similar reactions to pigment movement in their pigmented cells. The physiological control of photophore luminescence supports the evolutionary theory that this control was selected early in the evolution of lantern sharks.
Dwarf lantern sharks, or Etmopterus splendidus, belong to the family Etmopteridae, which contains many species that share the ability to emit light via bioluminescence. Bioluminescence is speculated to potentially have great ecological implications, but is one of the least investigated aspects of shark biology. This research presents the first morphological and experimental data on the workings of luminescence of this rare pelagic dwarf shark and potentially further insight into the ecological involvement of bioluminescence in sharks.